Okay, we'll move on to our fourth presentation. It's titled, Design of a Superior Factor VIIIa Mimetic by Coupling a Membrane Binding Domain to a Factor IXa Binding Antibody Fragment. This is from Rajesh Kumar and colleagues at the Children's Hospital of Philadelphia and the University of Pennsylvania.
Thank you. Good morning, everyone. So, as most of you know, that emicizumab is a factor VIII mimetic, which is used for the treatment of hemophilia A. This is a bispecific monoclonal antibody that has a 9A specific arm, binds to 9A, and it also has a factor X specific arm, binds to factor X. And original reports show that mimetic activity of this bispecific antibody is dependent on simultaneous binding to both factor IXa and factor X. And these original reports also specify that if we take monospecific one-arm antibodies, whether derived from factor IXa arm or factor X arm of emicizumab, they do not show any detectable mimetic activity. In contrast, some of the recent work has shown that if we take monospecific one-arm antibodies or their homodimer from anti-factor IXa arm of emicizumab, we see some mimetic activity. And that's exactly what we found when we made this single-chain antibody fragment VH9VL from 9A binding arm of emicizumab. So this single-chain antibody fragment shortened the APTT time in factor VIII deficient plasma, which is shown here. And this cofactor activity for this single-chain antibody fragment was weaker as compared to emicizumab, so that was expected. And both of them are several fold weaker as compared to factor VIII, which is shown here in the inset. So we tested binding of this antibody fragment to factor IXa using biolayer interferometry experiment, and it showed a KD of about 500 nanomolar, which is comparable to what has been reported for emicizumab binding to IXa, which is about 1 to 1.5 micromolar. So we know that emicizumab works by approximating factor IXa and factor X, which is one of the potential functions of factor VIIIa in the intrinsic tenace complex. The other function of factor VIIIa in the intrinsic tenace complex is binding to membrane and anchoring IXa to the membrane surface, and that can lead to enhanced factor X generation. So we thought that if we can somehow incorporate this membrane binding feature in our SCFV, we can see an increase in cofactor activity. So for that, we decided to fuse factor V C2 domain. So factor V C1 and C2 domain, and similarly factor VIII C1 and C2 domain are responsible for membrane binding. And we decided to use factor V C2 domain and not factor VIII C2 domain because there are inhibitory antibodies formed against factor VIII C2 domain in some patient population. So this C2 domain from factor V was fused to this SCFV VH9VL via a flexible linker of about 5.7 nanometer. So the idea here is that once this factor V C2 domain in this fusion is bound to membrane, this can possibly orient this SCFV at the membrane surface to optimally engage factor IXa, and then this can then lead to an enhanced factor X activation. So since the framework sequences for SCFVs are very similar, and there are several structures for SCFVs known, and we also have crystal structure of factor V C2 domain, we were able to model the structure of this fusion protein. We recombinantly expressed and purified this protein from mammalian cell culture, and we did membrane binding experiment using synthetic phospholipid, using light scattering experiment, and it showed a KD of about half a micromolar. When we did this experiment in complex with factor IXa, we saw a much enhanced co-localization of these mimetics, this membrane binding mimetic as compared to the one that doesn't bind membrane. So how about cofactor mimetic activity? So once again, we did APTT with factor VIII deficient plasma. So this is just the SCFV, and we saw a substantial amplification in the cofactor mimetic activity in this SCFV C2 fusion. And what was surprising is that the cofactor mimetic activity of this fusion was even superior to the emicizumab, which is shown here in blue. So in order to fairly compare the mimetic activity of these variants, it's important to draw factor VIII equivalence, and that's what I'm showing here on right. So here we have factor VIII concentration or mimetic concentration on log x-axis, and APTD clot time in seconds on y-axis. And once again, this is factor VIII deficient plasma. So if we, for reference, if we take 0.3 nanomolar of factor VIII, that's about 30% of our physiological concentration of factor VIII. This gives us a clot time of about 32 seconds. So to achieve a similar clot time, we would need about 2 nanomolar of this SCFV C2 fusion, and about 20 nanomolar of emicizumab, so that's 10-fold higher, and about 45 nanomolar of this SCFV that doesn't bind membrane. So that's about 22-fold higher in the APDT experiment. And these results were replicated when we did this in vitro factor Xa generation assay using purified proteins. And as you can see here in this in vitro factor Xa generation, this SCFV C2 fusion showed much enhanced Xa generation as compared to emicizumab. So next, we wanted to see if this enhanced mimetic activity is mainly contributed by the C2 domain. For that, we use this inhibitory antibody fragment E9-SCFV that we have made from a monoclonal antibody that binds to factor V C2 domain, and it putatively interferes with the binding of factor Va to the membrane surface. And as you can see here, this inhibitory antibody reduced the formation of Xa in a dose-dependent manner, and this inhibition saturated at about 85% level when we use 4 molar excess of this inhibitory antibody as compared to what mimetic we have in this assay. So next, we wanted to see how they respond in a global hemostatic assay. For that, we used thrombin generation assay using either 11A as a trigger or low tissue factor as a trigger. And Dr. Francis Ayombil and our collaborator, Dr. Rodney Kamir's lab at CHOP, was kind enough to help me with use the TGS